Okay, so to begin today, we're going to talk just briefly about the Nernst equation and protein synthesis and meiosis and clinical cases from the first lecture from last week, and then we'll talk and focus in on acid-base questions because you'll definitely for sure get 2 questions on this exam. You might get 2 on the second exam, and you might get 2 on the third exam, so 6 points. And then we'll talk about innate immunity. We'll talk a little bit about all of the clinical lab values, go over just a couple other points about bacteria, and get into the nitty-gritty of immunity and stuff like that. So first off, for the Nernst equation, again, I showed this last week, but again, just have that equation on your cheat sheet, and it's literally going to be a plug and chug, so make sure to bring your calculators. So a cell has an internal potassium concentration of 112 and external concentration of 4.2. So remember, the equation is 1 over the charge, so +1 for potassium, +2 for calcium, things like that. Chloride is a -1. Anyways, times 61 times log of 4.2 over 112, that gives us -87. And that would just be a very straightforward way to answer the question, and then you can move on. Again, I'll also send you all of these slides. And if you guys aren't usually in my group, just shoot me an email so I remember to add you to my list. Protein synthesis. So for protein synthesis, it's basically DNA goes to RNA goes to protein. So remember, DNA is kind of like the cookbook where you have all of your genomic recipes for proteins. So you have a DNA sequence, double strand, then you transcribe it into messenger RNA, and that messenger RNA we have to translate into protein. So you'll have to grab a chair, I think, on the outside.
Okay, I don't care.
Okay, that's fine. Okay, so remember, on the mRNA, you have sequences of nucleotide bases, and so every 3 sequences we call a codon, and that codon has a corresponding anticodon, and that anticodon is what's connected to the tRNA, transfer RNA, and the tRNA is what we have up here. So you see that it's got the anticodon which binds to the mRNA sequence, okay, so they match up, and each tRNA carries a corresponding amino acid, okay. So by having these anticodons matching with the codons, we have an amino acid that chains together with all of the other amino acids, and eventually we finish reading the sequence up to a stop codon, and that stop codon will tell us, okay, the protein is done, stop with these polypeptides, and then the protein sequence can go have those post-translational modifications like in the ER or the Golgi or wherever it's destined to go. Okay, so that's just protein synthesis. Is everyone kind of familiar with this now? Okay. Stages of meiosis. So with meiosis, remember it's we've got 2 sets of mitosis basically. So we have prophase, metaphase, anaphase, telophase. In prophase 1, that's where females have all of their eggs stopped at. So that's all in utero. So before you're born, and all of your eggs are basically at prophase 1. Once you hit puberty, the eggs once a month get ovulated to, and then they go through the sequence up until metaphase 2, and they stop there. And unless they're fertilized by the sperm, then they'll finish through telophase 2. Otherwise, they just end at metaphase 2, and that's it. So remember, so females, they start with all of the eggs that they'll have throughout their lifetime, and then they go through what's called atresia. And so each month you lose like a certain number of eggs, and you only ovulate 1. That 1 again goes from prophase 1 to metaphase 2, stops there. If fertilized, then you finish meiosis, and then you begin the fertilization process. But if not, it just ends there. Okay. So for the test, I don't think he'll ask you too many specifics about meiosis, like what phase is this versus that. But good to be familiar with the process. Okay, so again, this is another clinical case. So this is the same example I showed last week, but remember the first two letters, KG or whatever it might be, those are the initials of the patient, so you don't need to record that. But for your cheat sheet, it's good to know the age of the person, the sex. So I changed it to like 54M, 30-year history of smoking. That's also important. So you can write, I wrote HX of smoking. That's good to know. And then you can have things like he gained weight, he had these lab values. And if you don't want to include specific numbers, but if you include things like he had increased creatinine, increased BUN, things like that, those are important. Okay. So this is what the corresponding question would be. So a 54-year-old male with a 30-year pack year history of smoking and small cell lung cancer presents to the oncology outpatient clinic for lab work. Lab results show which of the following is the most likely cause. So if we look back, it's literally the exact same numbers, right? And if you record it on your cheat sheet that it was SI-ADH, which is syndrome of inappropriate ADH secretion, then you know here that the answer is ADH. Yes.
Sorry, you may have covered this last week, but so are the case studies that he's given us to look at going to be literally exactly the same?
It's going to be like cut down from like that huge paragraph, but it's going to have like those key things that I just told you to highlight in pretty much the same.
So if you have them on your cheat sheet,
you're all set. And make sure to look at all of the ones in section 1 on his website, okay? So all of the ones in section 1. Hi. Yes.
So do you recommend putting all of it, like a shortened version of all of them on our cheat sheet somewhere?
Yes.
Yes, okay.
Yes. Try to fit them. And again, size 4 font works, and I would recommend if you do use color, use it sparingly and like very organizedly. So just make sure you know where everything is on your cheat sheet, okay? So now we'll start talking about the actual lab medicine PowerPoint. So with fluid movement, there's a lot of different filtration kind of things, but the main takeaway is that you have either hydrostatic pressure or you have oncotic pressure. So hydrostatic pressure is kind of like pushing away pressure, whereas oncotic is like pulling in, okay? So at the arterial end of your blood vessel, for example, like let's talk about capillaries, you will push out fluid based on the hydrostatic pressure being greater than the oncotic pull of kind of your tissue area. And then for the venule side, you want to pull fluid back in, and so you have a higher oncotic pressure than hydrostatic pressure at that end. And then any excesses, they drain out to the lymphatic system, okay? So this concept is more like a big picture idea that when you have higher pressure on one end, it goes to areas of low pressure. And if you have like higher pushing pressure, you obviously will push out. And if you have higher pulling pressure, you reabsorb in, okay? So another big idea is the idea of osmosis. So with osmosis, again, the higher concentrations of water will move to areas of low concentration of water. So you want to make sure if someone tells you something is hyperosmotic, that's referring to the water content. But if someone is telling you about the solutes, okay, hypertonic solutions, then that means it has less water because it had more solute in proportion, okay? So those are key distinctions to make. So always think in terms of water, and water will move towards areas of less water to balance everything out, okay? And if it's like too much water is going in, the cell will swell, the cell can burst, and if it's too much water going out, the cell will start to shrivel up, okay? This is, I think, an important slide or figure to include on your cheat sheet. It's the edema picture. So what it's telling you is if, for example, you had increased capillary permeability, like for example from a burn or allergic reaction, you start to lose plasma proteins. So plasma proteins are important for this oncotic pressure, okay? So there's a difference between your capillary pressure and your tissue pressure, okay? So you're losing plasma proteins and you're increasing the oncotic pressure of your tissues, so your tissues want to pull in more fluid, but as they pull in more fluid, that's like an increase in fluid, which is by definition edema, okay? When you pull in that extra fluid, you start to have all of these other problems like lymph obstruction, for example. Like too much fluid causes other problems, okay? If we had some form of salt water retention problem or heart failure, we could have fluid movement into tissue also leading to edema, okay? And for example, if we talk about cirrhosis or malnutrition, so again, we lost plasma protein, but that contributes to the capillary oncotic pressure, so we're not filtering out as much fluid, so we're not reabsorbing as much fluid. So all of these factors somehow lead to edema in one way or another, and the main concept is, just know edema is buildup of fluid, and so if he ever asks you a question related to this, you can kind of reason it out that edema is something somehow involved. So this is a figure of ADH secretion. So ADH, antidiuretic hormone. So this is to, overall, if you have too much fluid, first if you have too little fluid, you want to increase your fluid, right? So I increase my thirst because I want to, make the body drink more. I also increase ADH secretion to make sure I secrete less, okay? So if I secrete less, I lose less water, check. If I lose less water, that means I'm retaining more water, check. If I retain more water, it circulates throughout the body, and once it circulates throughout the body, it starts to balance out the plasma osmolarity, okay? So osmolarity, again, is kind of a measure of the the solute concentration of your fluid. So you always want to keep that around like 300 milliosmolar, okay? And so all of this for ADH secretion is somehow to make sure your osmolarity goes back to normal, okay? So you're no longer, so once this all completes and you've gained enough fluid, then you don't need to secrete any more ADH because we don't need to retain any more water, and you're not thirsty anymore, okay? So this process is constantly happening. Another system is called the renin-angiotensin-aldosterone system. And so overall, this is another way to incorporate more fluid into your system. So if my kidneys, okay, hence the renal component of this, if the kidneys sense there's a decrease in blood pressure, okay, then they start to release what's called renin. Renin gets into the bloodstream and activates angiotensinogen, okay? Angiotensinogen will be converted into angiotensin 1, and then we have this enzyme in the lungs called ACE, angiotensin converting enzyme, which will convert Ang1 to Ang2. Ang2 can then act on the adrenal cortex to release aldosterone. So the adrenal cortex, again, is right above the kidneys. So right above the kidneys, you start to release aldosterone. And aldosterone will make you keep sodium. And if you keep sodium, that means the system has less water, right? So then the water from your that could have been excreted, it wants to be retained to like balance out that increase of sodium in the system. And so when you keep more water in the system, then the blood pressure starts to even out, and then you can kind of stop this feedback loop, okay? So like, as long as you have that decrease in blood pressure relative to the kidney area, then they keep secreting renin. But once it balances out, once you get that aldosterone into the system and you start retaining water, then you can stop, okay? But the kind of exact opposite system of that is ANP, atrial natriuretic peptide. And so what this is if I have too much sodium, I want to get rid of it, okay? So if I have too much sodium, I either want to get a lot of water in my system or somehow like get the sodium out, right? So I release, so my atrial cells, they start kind of stretching because they realize like, oh, there's more plasma volume being kind of uptaken here. And you start to release this atrial natriuretic peptide. So we, first we can have decreased reabsorption of sodium. Okay, so we don't want to keep sodium because we have too much sodium. Then we decrease the renin-angiotensin-aldosterone system because aldosterone is what makes us hold on to the sodium. And lastly, we tell the glomerulus to increase GFR, which is the filtration rate, because the more we filter, the more kind of solute we get rid of, okay? So all of these things are to get rid of if we have too much sodium, natriuretic, okay? Moving on, talking about potassium and calcium on membrane excitability. So if in the middle, let's focus here first. So this is telling us about potassium levels. And potassium levels will help us dictate kind of the starting resting membrane potential. So if we have a low potassium count, remember that means we have less, we're less positive, okay? And if we're less positive, we're more negative. That's where we are here. And if we're more negative, it's even farther to hit this, let's say, -60 threshold for membrane excitability. So it's even more difficult to get excited and start an action. So if we have low potassium, harder to start an action. If we have high potassium, then we're much closer to that threshold, okay, because we're more positive, less negative, okay? Much closer to the threshold, so easily able to get excited and cause an action. On the other side, on the flip side, we have calcium levels. Calcium shifts kind of the where the threshold is itself. So if we have high calcium, our threshold is farther away. If the threshold is farther away, it's harder for us to get excited. But on the other hand, if we have low calcium, the threshold gets pulled down. It's closer to resting membrane potential, so it's easier to get excited. So if we have low potassium or high calcium, those two relate to less excitability. And if we have high potassium with low calcium, then that's increased excitability. It's easier to hit the threshold. Okay, threshold and resting membrane potential are closer. Yes?
I'm sorry, what do you mean it pushes the threshold away?
So like, the threshold, like, let's say it's -60, for example.
Is the threshold the white line?
Yeah.
Or, okay.
So like, in theory, like, it would be farther up. It'd be like -50 or -40.
Okay. Right.
You know, so then it's,
Oh, okay.
yeah.
I was confused why the white line was still there.
Yeah,
Okay.
yeah. Okay, any other questions? Good. So EKGs, you'll have an entire lecture on this for second unit, but I guess the biggest thing for now is, again, the changes in the serum levels of potassium and calcium affect the regular electrophysiology of your cells, okay? So you don't need to memorize like the PEQR, all that stuff until second section, okay? So just know potassium and calcium can affect how your cells function. Next we'll talk about calcium balance. So there's two conditions you can have. You can have too much calcium, hypercalcemia, or too little. If you don't have enough calcium, okay, so if we have parathyroid hormone, okay, PTH increases calcium through various mechanisms, okay? So PTH, if you use PTH, it will act on the intestines to absorb more calcium. It will also act on the kidneys to absorb more calcium, and also act on the bones to like kind of break apart the bones a little bit to release more calcium. Okay, so PTH will try to increase calcium levels. Okay, so that's in conditions of low calcium. Hypocalcemia, so we increase PTH, we activate vitamin D in the kidneys, and then we absorb it in the intestines, reabsorb it in exchange for phosphate in the kidneys, and we also break up bone for calcium. The breaking up bone part is kind of like the final, like last straw if you really don't have any calcium. Otherwise, it's more the intestines or kidneys. On the other hand, if you do have too much calcium, you don't need PTH at all. So It's just the opposite of having too little, okay? So acid-base. So this is kind of where it gets tricky. So this is the part that I would kind of focus on from this PowerPoint, definitely, because you'll be asked at least 6 questions throughout the next, like, 3, for all of your exams, like, total. You'll have at least 6 points towards this stuff. So carbon dioxide plus water makes a component H2CO3, which instantly will dissociate into H plus and HCO3 minus, okay? So these are all in equilibrium always, and they just kind of go back and forth. But the way you can think about it is like the more carbon dioxide, CO2, in your system, the more H plus you'll have in your system, okay? The more, and that's kind of like the biggest thing. H plus and CO2 levels, let's just kind of roughly say they're proportional, okay? Roughly. So these normal values of pH, CO2, and HCO3 minus, you definitely should know. So it's 7.4 for your pH, 40 for your carbon dioxide, and 24 for your HCO3 minus, okay? So there's different ways we can go about regulating all this acid-base stuff, either in the lungs or in the kidneys, okay? So in the lungs, we control carbon dioxide. The more you breathe out, the less H plus is kept in the system, because the carbon dioxide, again, will become H plus, so less carbon dioxide, less H plus. Make sense? Or we can work with the kidneys. The kidneys can, like, pee out H plus, okay? They'll secrete it out, okay? So putting those two together, you can also reabsorb bicarbonate, but it's not as relevant as your, like, H plus secretion. So let's think about this graph. I think this is a very good graph to look at and to understand from all different ways. So first off, if you ask someone a question about this kind of stuff, you first want to identify, are we talking about an acidic or a basic situation, okay? So is the pH above or below 7.4? So pH scales, remember, are logarithmic. So every 0.1 is actually a big difference, okay? Every 0.1 change is a very big difference, okay? So we're acidic if we're less than 7.4, but we're acidic if we have more H plus. Does that make sense? Mm-hmm. So because pH is negative log of H plus concentration, as the pH goes down, your H plus concentration is actually going up. So always keep that in mind. So we have low pH, that's acidic. If we're acidic, for example, and we have normal CO2 levels, that's not a respiratory problem. If it's not a respiratory problem, it has to be metabolic. So then, if we look along this curve, right, metabolic acidosis, so the pH is less than 7.4, and we're also along this 40 line on the graph. So do you guys all see this? So to read this graph, pH going like kind of vertically, like up and down, right? Then we have our CO2 lines is kind of like diagonally across, and then we have bicarb. So bicarb, you don't really need to look at. I think it'll confuse you if you think too much about it, okay? So if we're along the normal CO2 line of 40, then it's probably a metabolic problem. If we have too much CO2, right, then that probably means we have too much acid in the system, because again, too much CO2, too much H plus. Too much H plus, lower pH means we're acidic, okay? If we have less than 40 millimeters of mercury, then we have too little CO2, that means less H plus, that means higher pH, that means basic. Alkalosis, okay? So, and lastly, after looking at whether it's acidic or not, or respiratory or not, then we'll look at is there compensation or not. So is it a chronic problem or an acute problem? If it's acute, it's only lasted for a little bit, or a very short amount of time, there's not much compensation necessary. But if it's chronic, we might have some sort of metabolic or respiratory compensation. And so sometimes, if he asks you a question on the test, you might not get something that's like nicely fit into here or there. There might be a shift towards the center relative to like the starting point, but just reason it out. Like if I had a respiratory problem, I would compensate metabolically. If I had a metabolic problem, I would compensate with respiration, okay? So if I had a metabolic problem, I would try to breathe out more or breathe out less, okay? And that adjusts the H plus through my lungs. Otherwise, it's my H plus secretion that's affected for the metabolic compensation of a respiratory problem, okay? This is like pretty much the same thing that I just said. Also, this equation is good to have as well. pH equals 6.1 plus log of bicarb concentration over 0.03 times PCO2. Okay, it's good to have. This graph basically says what I said as well. You can review it on your own time. So, respiratory acidosis. We've gone over this. So, COPD or acute respiratory problems. So COPD, we can't breathe out as much. If we can't breathe out as much, that means we're retaining more CO2. If we retain more CO2, that means we have more H+. If we have more H+, that means we have a lower pH. If we have a lower pH, that means we're acidic. Putting that all together, it's a respiratory problem, and we're acidic, so respiratory acidosis. Does that make sense? Metabolic acidosis. So it could be a kidney failure, so the kidney can't secrete out the H+, so we build up H+ there. Could be diabetic ketoacidosis, or it could be long-standing diarrhea. So like losing lots of base in that case. But either way, you would compensate with your respiration. So you have a high buildup of H+ that you can't get rid of, so you breathe out more. So that's hyperventilation. Breathe out more CO2 to get rid of H+. So that's metabolic acidosis with some form of compensation. He might ask you anion gap. Anion gap is the Na concentration minus the chloride and the bicarbonate concentration. And if you have a high anion gap, it relates to like adding base to your system. If it's normal, then you lost base. High anion gap is adding acid. Normal is like losing base. I don't know if he'll ask you that, but it's a good equation to have.
What does the high and normal mean? Like, how does that?
I'm not too sure what the standard number is. Like, he might like give you an actual value, or I think it might be gone over later.
I think it was in the lab 20 or something.
Yeah, I'm pretty sure it's like there's like a range that's on one of the long lists, and we'll get to that soon. Metabolic alkalosis, so if, for example, I'm vomiting it out. When you vomit, you lose acid, right? Vomit is acidic, so you lost a lot of acid, so you're basic now, and it's a metabolic problem because we're not breathing. You know, you don't breathe vomit, you have a metabolic problem that causes that. So because it was a metabolic problem, you compensate with respiration, right? So you would, how would I compensate that? If I'm metabolically alkalotic, so I have too little acid in my system. So I want to keep as much CO2 in as possible. So I keep in more CO2 by breathing less. So hypoventilation. Okay? And lastly, respiratory alkalosis. So I can hyperventilate a lot, and when I breathe a lot, I lose a lot of CO2, and so my kidneys would just tell me, okay, stop secreting H+, or like retain HCO3 minus or something like that, okay? So that's kind of the breakdown of the 4 different categories. So remember, metabolic problems are along the normal CO2 line, respiratory problems are above and below the CO2 line, and then acidic versus basic, above or below 7.4 of pH. So let's try a problem. Prolonged vomiting. Anyone have a guess? So what does prolonged vomiting mean? 1, is it respiratory or is it metabolic? Metabolic. Okay, is it acidic or basic? Basic, because we are losing a lot of acid. Okay, so which one is a metabolic basic problem on this? A. Does everyone see that? So A is right up here. Okay. Chronic COPD. So COPD, 1, respiratory or metabolic? Respiratory. And then is it acidic or basic? Acidic, because we can't breathe out as much H+. So now we have a respiratory acidic problem. So which one is it? B, yeah, everyone see that? So right up here, okay. Alright, good. So for all of these panels, I would just like, oh yeah, anion gap, 8 to 16, that's your normal range. Yes.
What does that show you? Like, what is there anything they use that for specifically?
I don't think it's like super important.
That would like it wouldn't come up in
Yeah.
like the clinical case.
Right.
Or anything. Okay.
So I'm gonna just run through it like this. I'll send you this like in a nicer format than just in a PowerPoint slide, but this is kind of breaking it down. So sodium levels, if you have too much sodium, you might be dehydrated, or like you could have lost out on water, right? If you had too much potassium, it could indicate kidney failure. Too much, you could have a high chloride count if bicarbonate is low, because chloride serves as kind of like a buffer ion, and so it's serving as like the negative charge in your system. Bicarbonate relates to acid-base problems. BUN, that if it's high, it can also relate to kidney problems. Creatinine, same with kidney problems. Glucose, fasting glucose levels, if it's 100 to 128, that's, I think it's just like, you ate, and then greater than 128 is diabetes, like could be diabetic problems. And then moving down lower, calcium levels relates to cellular behavior, like are your cells doing okay? Like if you remember on the first lecture, we talked about the problems if you had too much calcium or too little calcium, and how that related to, what is it, cell death, irreversible cell death. So look at that slide again. Phosphate, PI, that can lead to calcium deposition, because if you remember back to parathyroid hormone, the more parathyroid hormone you have, the more calcium you have, but in turn you lost out on phosphate. Uric acid levels can relate to gout. ALT and AST can relate to liver problems. ALK, alkaline phosphate something, A's, can talk about the biliary duct or bone problems. GGT, biliary duct, bilirubin, liver stuff, because that's where you produce all of those necessary proteins and stuff in the liver. And same with albumin, okay? So albumin levels, it can be low if the total pressure, total protein is also low. That could be a kidney or liver problem. And then if it's just the normal albumin is low, that's just a liver problem. And total protein can be a liver or a kidney problem. LDH can relate to cellular necrosis. And so that was a lot. Whew. So this is what you'll get on an exam, though. It'll literally just be all these numbers, and it's like, what's wrong? And, you know, you just kind of like go through number by number,
Look at my cheat
like,
sheet.
what's wrong? You know, just look at your cheat sheet, line up numbers, and you're like, okay, this, check, 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 check. Okay, so we won't go through that because that'll take a while. And then, again, like all of these values, again, just throw it on your cheat sheet in case he asks you. Other than that, like, not too much. Also, remember, like, never let monkeys eat bananas. The order for, like, the neutrophils, lymphocytes, monocytes, eosinophils, basophils, okay? For a relative proportion of, like, how much there is in the cell, okay? So any general questions on lab medicine, not PowerPoint? Okay, so everyone's comfortable with acid-base now? Pretty much? Good? Good? Okay. All right, so let's talk about immunity. So bacteria, there's two main ways we distinguish bacteria first in the lab. We first look at gram-positive versus gram-negative. And so that relates to the peptidoglycan layer of the bacteria. So gram-positive stain because they have a thick peptidoglycan layer, whereas gram-negative ones have a very thin one, and they also have like an outer membrane that prevents the stain from being taken up, okay? They can be cocci, which are spherical, versus bacilli, bacillus, rods, okay? So they can like literally be circles or like long, stringy rods. They can rely on oxygen in different capacities, so aerobic microaerobic, facultative, or obligate. So obligate means they have to have oxygen. Facultative means they can kind of like deal with it, and then like micro means they don't need very much. They need low levels, and then aerobic is just the general sense. Toxic formation. So endotoxins are from gram-negative bacteria. So typically gram-negative bacteria are the ones where you get like more difficult diseases because you get these kinds of toxins. And that's released from cellular death. Exotoxins, on the other hand, are from gram-positive bacteria. And that's secreted from live bacteria. So an example would be botulinum toxin. So, and then you can have spores form, or they can be intracellular, meaning they infect the cell, versus they are like just in your bloodstream, extracellular. So in this graph, I recommend having it on your cheat sheet as well, but keep in mind that he did put like a switch here. So, and when you try to copy paste it onto your graph, it won't include these like sketchy boxes that he overlaid. So make sure you copy like those boxes over, or remember to like note it, okay? So yeah, and you'll notice if like it says rods, but then you see here that it's like clusters, yeah. But again, just remember to note the switch. Let's see, things to note. Clostridium comes up a lot, like Clostridium perfringens, if you guys remember. That was for like the gas gangrene. You might come across something called C. diff, Clostridium difficile. That's like a very difficult bacteria to get rid of. Listeria monocytogenes can cause other problems, like, and Neisseria can be gonorrhea, for example. I think Campylobacter can cause, it's called Guillain-Barré, and you'll see that a lot later, I think. But again, just like look through these names. Staphylococcus, Streptococcus, obviously those are big ones. Also, Staphylococcus is the most responsible for nosocomial infections. So like people in the hospital like tend to get staph infections. It's like it's very prevalent, and like people pick up like MRSA pretty easily because it's just kind of like everywhere. So staph isn't usually a problem for healthy people, but like for sicker people it can like really build up. Streptococcus causes strep throat, and you'll see later that it also relates to something called rheumatoid arthritis, and that's probably next section. Which of the following is the common cause of nosocomial infections? I just told you. Staphylococcus. Cool. Yeah. Viral infections. So viruses themselves are not living organisms. They are simply packets of genetic material, and so they are intracellular, I guess, things. So they have to live inside of a host cell that is living to reproduce. So they typically will get absorbed into the cell somehow. Sometimes they come with envelopes, sometimes, to varying degrees, and they kind of like spit out all of this genetic material, and they either bury into the genome itself, and they just stay there for a while and replicate as the cell replicates, or they kind of like hijack the cellular machinery and just start building viral proteins. So there's a lot of different ways that viruses work. You don't have to know too many details, just know that they like get into the cell, they can't live without a host, and that they can be DNA or RNA viruses. So viruses can do, so I guess for this one, we can say that does anyone have an answer? Yes. Number 3, they replicate their genetic material inside host cells. So they aren't killed by antimicrobials because they aren't living. They have to contain either DNA or RNA, kind of by definition, and they need to have the host's genetic, like protein machinery to build their own self. Like they need to replicate somehow, and so they don't carry anything but information, and then they hijack the cell's information. Okay. Medically relevant fungi, what's good to know is that Candida albicans are like your gut flora, and they can cause opportunistic infections. So if you're feeling kind of down, then like they're usually like in your gut already, and only if you're kind of down do they actually cause a problem for you. Okay, so like bacteria, a lot of the time, like we have tons and tons of bacteria in our system, and they're pretty nice to us unless we're like feeling horrible. That's when they kind of take over, or like someone else edges the nice ones out. Yes.
What does it mean by gut flora?
So like flora is kind of a term for like kind of like colonies and like clusters of bacteria. So they're normally inside like your intestinal tract, GI system, and so they just kind of like stay there, and they kind of help like degrade things, and they kind of feed off of bad things, you know, in there, and like degrade stuff. But they normally just reside there, not doing anything. Oh, sorry. Aspergillus is a respiratory infection. I'm sure you've heard of it. Tinea, so you can have Tinea capitis. That could be like a head kind of fungi. Curris, that can cause jock itch, or pedis for athlete's foot. And then there's two types of forms of fungi. So there can be mold, which is more filamentous, and there's yeast, which are more spherical. Okay? That's just simple what you need to know. So the innate immune system now, okay? Overview, so there's either your humoral or your cell-mediated types of immunity. So humoral immunity is more like extracellular. It resides in the bloodstream, so your white blood cells and your immune system can like come into contact with the pathogen and say like, I will kill you now. Whereas your cell-mediated problem, that infects the cell itself, so it tends to be a little bit trickier to detect. Because your cell will have to be like, first off, are you one of us or not one of us? And if you are one of us, are you like a poser, or are you like somehow infected? And then you can kind of proceed with killing the infection. So you can be have an innate system with your myeloid cells, or you can have adaptive with your B cells. B cells relate to antibodies. And myeloid cells are like your macrophages and more like non-specific kind of things, like neutrophils. Cell-mediated will relate to natural killer cells. So natural killer cells are innate, but they still relate to cell-mediated, so they check whether or not the cell itself is infected with something, okay? And then T cells, they have to have what's called this MHC, so membrane histocompatibility complex, or major histocompatibility complex, that binds with a receptor to check whether or not the cell is like good or bad. Okay, so they check inside the cell basically, like looking whether your cell is infected, versus a B cell, which is like, I see a pathogen, like one of my arms made contact with you, so you're probably bad. Okay, and I'll explain that soon. So cells of the innate immunity, you have a ton, but we'll focus on macrophages, neutrophils, eosinophils. Okay, those are pretty much your big ones. So typically, if you have a bacteria, let's say, floating around in your bloodstream, your macrophages will tend to see it, and they can release what are called cytokines or chemokines, kind of the same thing. So they release these things into the bloodstream, which tells all of your other parts of the immune system, like, hey, we need more like backup, okay? We need backup to kill this guy that I just saw that shouldn't be here. So because these chemokines, how they work, it'll tell your blood vessels to dilate, because it's like, let's get a bunch of blood over to this area, because more blood, more of our good guys will come save us. And so you get inflammation in this area, and you start to get a bunch of other helpers to come into contact with the bacteria. Phagocytosis is more with, so the process of phagocytosis and how like neutrophils work. So neutrophils, let's say they sensed that there were chemokines in this area. So we've got kind of our cellular layer, and we've got like our, the wound where we have it. And so the neutrophils diagrammed here, what's called pavementing occurs. So the neutrophils get up to the vessel wall, the blood vessel wall, and they're like, okay, like we're ready to see what's going on. So they have to, so your blood vessel has to kind of retract the endothelium a little bit, so to give space for the neutrophils to go through. So you increase vascular permeability, and those neutrophils can now like squeeze through between the epithelial layers of those blood vessels, and then it's called diapedesis, where they move through towards the they move through the junctions. Okay, so they squeeze through after your endothelial retraction, and now they go towards, so chemotaxis, going towards the highest concentration of where all these chemokines are, chemotactic factors. So pavementing, they get up close, they're like, okay, where's the problem? Endothelial retraction, your blood vessels start to like move apart a little bit, or like the cells will start to separate so your other helpers can get through. Diapedesis, so they actually are going through, and then chemotaxis, where they are going, they go to the highest concentration of these chemotactic factors. Okay. Let's see. So adaptive immunity. So we have lymphocytes, and so all of your immune cells, they come from some sort of what's called a progenitor, and it'll split up into different kinds of cells, okay? So in your bone marrow, from your bone marrow, you get B cells or you get natural killer cells, but in the thymus, you get T cells. And then from there, everything kind of differentiates more, okay? So it's important to remember that the thymus is where T cells are produced, and bone marrow is like all of your other immune stuff, okay? But basically, let's focus with T cells first. So your T cells, they can either become regulatory, cytotoxic, or memory or helper T cells, whereas your B cells are either plasma cells. Plasma cells are the ones secreting antibodies, or they're memory B cells, so they are just for storage. So there's either those more like active kind of immune cells versus the memory cells. So memory cells, they stay alive for a very, very long time in case a recurring infection occurs, and if that does happen, then your secondary or primary response to that new infection is a lot quicker, because somehow one of your cells remembered that you've fought this before, and they have the tools necessary to combat it much easier, okay? All right. Structure of an antibody, so imagine like a Y shape, though it's not actually like this, it's more like this crystalline structure that happens to be in a Y, but you have a constant region, that's the Fc, okay, and then you have the variable regions, and so this picture is a little bit misleading in that it says like light chain is up here, heavy chain is down here. So like technically both components have like the heavy and light chain, but I guess for the purposes of this portion we can kind of think of it as like variable region is at the top, heavy constant region is at the bottom, okay? Alright, so there's different kinds of immunoglobulins, so these antibodies, okay? So we can have IgA, and that's secreted through some form. They deal with more like epithelial or secretory cells. So you find them around the GI, broncho, respiratory, like lung area, genital, urinary areas. There's all sorts of secretions in all of those areas. IgE are for mast cells, and those you'll find are most related to parasitic worm infections that you'll see for this class. IgG is kind of the starting point for all of them, so it's more general, it's just kind of like in the blood. IgM is the first guy, like everyone gets IgM, and then IgM can do a class switch into A, G, or E. And then D, we don't really know what it does, but it's just there, okay? So these are the different types of immunoglobulins depending on like where they're most useful or what they're most used for, okay? So IgM is what everyone starts with, and then depending on the situation, IgM can kind of like change, class switch into A, G, or E, depending on the situation, okay? So IgA is the one that you get from your mother. I'll send like I think I deleted a slide about that. But it's basically like, because a fetus doesn't produce its own immune system, it doesn't have its own immune system, it gets it from its mother. Okay, so IgA is just important. Let's see. So these are the different ways antibodies can work. So one, you can block receptor sites. So that's like you have the antigen and you can like bind to the whatever that's floating around. It could be a toxin, could be like a protein, could be whatever. Just blocks it. So it can't bind to anything else. Two, it could bind to a bacteria, and if it binds to a bacteria, it could be like a tag. And your macrophage has non-specific receptors for the constant region of an antibody. So it's like, oh, an antibody bound to something. I'm going to grab onto that and then eat everything with it, okay? It can block a virus, it can block bacteria, it can block toxins, okay? So they complement it and neutralize it. You can also be a tag for a mast cell, and a mast cell will degranulate lots of factors over a parasitic worm in case 4. Or it could be for number 5, like you have these antibodies and then like this thing called membrane attack complex forms, MAC, and then it literally punches a hole through the bacteria. So many different ways that antibodies can work, okay? So B cell fundamentals. So B cells, like, how are we supposed to know like what does and does not constitute a self-antigen? How are we supposed to know like all the different combinations of possible antigens out there? So B cells, what they do is your DNA will randomly differentiate through a process called VDJ recombination. And if you guys are human science majors and taking immunology later on, you'll learn about it. But basically, it's like a series of random recombination of your genetic material so that you produce like billions and billions of different combinations. You can have like 10 to the 13th combinations plus more. And so all of those are screened somehow. So your what happens is a nurse cell will present an antigen to the antibody that's forming, okay? And if the antigen reacts to it, if it reacts to a self-antigen, you kill it off immediately, and that's called negative selection. So you want to prevent anything that reacts to yourself from getting out into the bloodstream where it can react, okay? If you've screened through all of your self-antigens, then you can move on to the bloodstream and you can find your like antigen and really mature, okay? If you don't find your antigen, you don't really do much, you just kind of degrade away. But replication, the process I just described is called somatic hypermutation, so that's how you make like that 10 to the 13 or 10 to the 21 like copies. It's just like many copies. Okay. Let's see. So you think this is a diagram of the parasitic worm. So let's say a parasitic worm came, and so what happens? The worm comes and it has some antigen floating around. So something that describes, oh, a worm is in the body. Then the antigen like gets picked up by a B cell that happens to have a receptor for it, and it's like, oh, the antigen for a worm is in the body. I should produce IgE antibodies to combat it. The IgE antibodies will bind to the parasitic antigen, and a mast cell will pick up the fact that the IgE are in the system and binding to these antigens. The mast cell secretes this ECF factor that like calls over eosinophils, and eosinophils, like through this similar process as the neutrophils, like from the blood like to your tissue area or wherever the parasite is, they'll come over and they will start to degranulate all over the worm with lethal enzymes and stuff like that. So if we're talking about how this process all works, we go from 1, so local antigen presenting cells present a worm antigen to helper T cells, for example. From there, we have a naive B cell binds to a worm antigen, it activates. Next, plasma cells produce IgE specific So the B cell, so a naive B cell finds its antigen, it matures and becomes a plasma cell. The plasma cell produces IgE, and the IgE binds to a mast cell. The worm antigen that's bound activates those mast cells, and the mast cells degranulate and releases ECF and histamine. That calls over eosinophils, which is what extravasate means. It means like the eosinophils are like coming through, and then the eosinophils will follow the gradient of the ECF and they'll find the worm. Once you find the worm, you find that the IgE are bound to the worm, and then you start to degranulate and then you kill the worm. So I would recommend, this is like a test question. I placed it in the correct order for you, but you know, like running through this a couple times just to make sense of everything. Part 1 isn't as important, but like 6 through 9, like in this order, are more important, I guess, for this, okay? Next, we have Peyer's patches. So that's gut-associated lymphoid tissue, commonly called gout, okay? So if you ever see gout or mouth, those will talk about some form of lymphoid tissue, okay? So what happens is you have your naive immune cells swimming around the Peyer's patches, and they're like, oh, I need to find an antigen before I can mature. Peyer's patches are like great places to find antigens because like there's a lot of transcytosis, so stuff gets pulled in from one end and thrown out the other, and so you like release all these antigens around, and you're like, here, naive B and T cells, go find an antigen, and then you can mature, and then you can go forth and like do stuff in the system. Yes.
Sorry. On the picture before, is it? Oh, sorry, the one you with the lumen of the intestine. Sorry. There's no B and T cells that are actually inside the lumen of the intestine. So basically, it's just like in the patch. Things are kind of, I don't know, by random maybe coming in, and that's like, you know, you're checking it out, and if you do find the antigen that matches, that's why it tells it sends HEA into the lumen. You couldn't do that just directly because there are no cells within the actual lumen.
So your immune cells are constantly circulating between different Peyer's patches around the body, and like they might find something here, they might find something there, like, or lymph nodes, they also go around lymph nodes to find antigens and stuff like that. But I think like that's starting to go into too much like immunology detail. But I guess like the biggest idea just to know is like your B and T cells, like once they find their antigen, they, you know, produce antibodies, or like your T cells will somehow like cause cell death, and we'll get to T cells in a moment on how they cause cell death. Great. Actually, I think I said IgA earlier. I meant IgG for the maternal circulation. Yeah, so that's a correction. And what's called so maternal circulation gives IgG to the fetus as the antibodies cross the placental syncytiotrophoblasts, which is like that kind of membrane or like crossing area between mom and fetus's circulation. Okay. So the mom's IgG is in the circulation, and once the child is born, he or she can start producing their own IgG, and eventually, after about 6 months, they have more of their own immune cells than their mom's. Okay. Cell-mediated response, T cells. T cells are different than B cells. One, because you don't have a free-floating T cell receptor. Okay, so a B cell receptor is an antibody. A T cell receptor does not exist floating around. Okay. B cells randomly like look around, and if they bump into an antigen, they can activate. But a T cell has to have a major histocompatibility complex with an antigen-presenting cell presenting an antigen on the MHC to it. Okay. So there's different types of APCs, antigen-presenting cells. You can have dendritic cells, you can have macrophages, you can have like mast cells, etc. They have an MHC on it, which can hold the antigen, and they give it to the T cell. So you can have a class 1 MHC, and that's for self-antigens, and that relates to CD8. CD8 means clusters of differentiation 8. It's just a marker. So CD8 relates to cytotoxic T cells. Cytotoxic T cells deal with self-antigen, so your own cells have been infected somehow, versus class 2, which is CD4 T helper cells that notices that a foreign substance has come into the cell. And again, T cells deal with intracellular infections, and they're a part of the adaptive immunity, okay? So if we look here, a T helper cell gets presented a cell. Okay, let's look at the left side first. CD8, MHC. Our cell is infected by a virus, but it is our self-antigen that is presented to the cytotoxic T cell, and the cytotoxic T cell realizes, like, hey, something is wrong with our self, so we kill it. The T helper cell, on the other hand, will be like, oh, it's a bacteria that is inside the cell, but it's the bacteria's antigen, which is foreign, that makes the T helper cell realize, like, oh, it shouldn't be here. So that's class 2, okay? So that's kind of the difference. It's very slight, but MHC class 1, self, CD8, cytotoxic T cells. MHC class 2 deals with CD4 T helper cells, deals with foreign antigen. Okay. T cells develop in the thymus. You have two types of selection, positive and negative. Positive selection means, can you interact with an MHC or not? Negative selection means, like, if you do interact, you bind too tightly, or you don't want self-reactive T cells, basically. So if you bind too tightly, we don't want you, okay? And you have immune privileged sites, like placenta, testes, ovaries, eyes, brain, thymus, because for obvious reasons, like, you don't want your own immune system, like, killing yourself in these areas. Like, you could attack, like, the sexual reproduction, eyes, brain, like, that would be very bad for further consequences. Lymph nodes are areas where you have tons and tons of antigen floating around, so B and T cells find a lot of drainage. So if we remember earlier about the fluid drainage, excess fluid drains into the lymphatic system. So this is where our B and T cells can find extra stuff floating around. Maybe they get activated, maybe they don't. And then, let's see how many more slides. Okay, 2 more. Ah, shoot. Okay, that was weird.
On the master.
Mouse doesn't work. Okay, let's end here then.
Thank you, Victor.
Welcome to
Thank you.
VoiceOver. VoiceOver speaks descriptions of items on